When you receive GenoSensor's Genetics of Taste kit, it will consist of two major components. First, a clear container containing temperature-stable components. Second, an ice box that contains temperature-sensitive components. The clear container contains all the components necessary for DNA collection and isolation. Solution A contains the necessary reagents for cell lysis and DNA extraction. Solution B contains a neutralizing agent that is used to prepare the sample for DNA isolation and PCR. Also included are taste strips and cotton swabs used for collecting cell samples. Inside the icebox, you will find an ice pack used for keeping its contents cool. Beneath that, you will find an aluminum bag that contains all the reagents necessary for PCR, including a PCR master mix, positive and negative controls, restriction digest master mix, as well as a DNA ladder for gel electrophoresis. It is important upon receiving the kit, you place the aluminum bag in freezer storage at -20 degrees Celsius. Before you begin your experiment, you should prepare your heat block, or water bath if a heat block is unavailable. Turn your heat block on and add water, or other appropriate materials to maximize heat transfer. Before using the heat block, be sure that the temperature sits stably at 95 degrees Celsius. Remove the PCR master mix from the aluminum bag and place it on ice to thaw. Once it is thawed, it is vital that before opening the tube you spin it down for 10 seconds, vortex it, and then spin it down again. Be sure to keep the master mix on ice until it is ready to be used. Begin the experiment by adding 200 microliters of solution A to a labeled microcentrifuge tube. Then, using the provided cotton swab, swab the inside of your cheek for 10 seconds. Place the cotton swab inside the tube and then cut it so it will close tightly. Then vortex the tube and place it on a prepared heat block for 5 minutes. Load the sample into a microcentrifuge and spin it down for 10 seconds to remove condensation. Afterwards, remove the cotton swab from the tube using tweezers. Be sure to wash the tweezers with ethanol between samples to avoid contamination. Next, add 20 microliters of solution B to the tube to neutralize the mixture and prepare for DNA isolation. Vortex the tube to mix the solution, then place it in a microcentrifuge at 12,000 RPMs for 1 minute. After spinning, a small pellet containing cellular debris may be visible at the bottom of the tube under the hinge. At this point, if you haven't already, prepare the master mix as explained earlier by spinning, vortexing, and spinning again. Begin PCR preparation by adding 10 microliters of the PCR master mix to a labeled PCR tube. Do the same for tubes labeled for positive and negative controls. Add 2 microliters of the sample supernatant to the appropriate PCR tube. Use the pipette to mix the sample in by pipetting up and down. If you do not immediately load the tubes into a thermocycler, be sure to store them on ice. Otherwise, load the tubes and set the thermocycler. At this point, for classes with shorter time periods, you may unload the PCR samples and store them in a cool environment for a later lab period. In this section, we will start digestion. At this time, we are going to open the plastic bag containing the 2x restriction digest master mix tube and thaw it by warming with your hand, then placing it on ice. Before opening the tube, spin for 10 seconds at 6,000 RPM, vortex for 10 seconds, and spin again. First, take 10 microliters of PCR product made previously and transfer it to a new marked tube, and then add 10 microliters of the 2x restriction digest master mix and mix by pipetting up and down. Incubate the final mixture at 75 degrees Celsius for at least 40 minutes or longer. Load the samples on the gel and proceed to gel electrophoresis or store at -20 degrees Celsius for later use. Running the resulting digested samples through gel electrophoresis, follow standard procedures with a 3% agarose gel with a DNA dye such as gel red or CyberSafe. It's best to keep the gel in the dark during the cooling and gelation process in preparing the agarose gel as well as when running the gel. Once the gel has run, view it under UV lamp. The gel image shows the various genotypes of homozygotes or heterozygotes correlated with the phenotypes of tasters or non-tasters. Please visit genosensorcorp.com for more information.